Good morning, ladies and gentlemen. First, I would like to thank the organizers for inviting me and for the possibility to discuss the role of regulatory RNAs in CLL biology. And we have recently learned, largely thanks to the ENCODE project, that over 3/4 of our genome is transcribed into RNA, while only a few % of that is actually used to produce proteins. And the rest of the RNAs acts as regulatory molecules. And the largest class is called long non-coding RNAs, and there is currently over 200,000 annotated long non-coding RNA genes in human genomes, so 10 times more than coding genes. And these molecules can regulate various functions such as gene transcription, chromatin structure, or bind and regulate other RNA molecules. On the other hand, there is a relatively smaller group of short non-coding RNAs, mainly microRNAs, that have a comparison to long non-coding RNAs, a simple mechanism. They regulate translation and stability of mRNAs with one complication, that one microRNA can actually regulate dozens to hundreds of different targets. And when it comes to long non-coding RNAs, there have been several reports in CLL comparing the expression of these molecules in healthy B cells compared to CLL, in indolent or progressive CLL. And we have learned that there are differences in the expression, and also that p53 can activate a number of these molecules. But frankly, when it comes to functional understanding, I think we don't know what are these molecules doing in CLL biology. So from now on, I will mainly talk about microRNAs where we have a bit better knowledge. And I'll start by mentioning the work of Carlo Croce, describing that the miR15 and 16 is deleted in this enigmatic 13q region of CLL. And this work was followed by a mouse model from Della Favera's lab showing that the deletion of these two microRNAs leads to a CLL-like disease in mice. However, this work also showed that a larger deletion that contains the host gene of miR15-16, which is actually a long non-coding RNA called DLO2, leads to a disease which is more aggressive and in a higher penetrance of this disease. So it actually suggests that this long non-coding RNA has additional functions besides encoding miR15-16. So for miR15 and 16, several interesting targets have been identified that are relevant for CLL, such as MCL1, BCL2, cyclin molecules, and ROR1. For DLO2, mostly we don't know, but there is an interesting report suggesting that maybe it acts regulating in CIS some neighboring genes involved in NF-kappa B activity. And also, there is accumulating evidence that there are actually two promoters, each regulating microRNA or the DLO2, so that actually these two can be partially regulated separately. miR15 and 16 is not an exception. So there is other 4 or 5 models showing that manipulation of a single microRNA can lead to a frank B-cell malignancy, including a CLL-like phenotype, such as the miR1792. And these studies were complemented by analysis of methylation and microRNA expression in normal B cells and in CLL subtypes, showing that there is differential methylation and differential expression of a number of microRNAs. However, for most of them, we still don't know what's their molecular function. So what I would like to do next is to show you 4 examples. Those are the microRNAs highlighted in orange that are both differentially methylated and differentially expressed, and how they regulate microenvironmental interactions in CLL, especially BCR signaling. And the first is the miR150 that we selected with Tom Kipps as a candidate, mainly because its low levels are strongly and independently associated with shorter survival in CLL, and also because patients that are more responsive to BCR ligation with anti-IgM have low levels of this microRNA. And we aimed to identify novel targets of this microRNA, none were known in CLL, which is still a technically challenging task because each microRNA can regulate a number of molecules. And we used an approach of profiling gene expression in CLL cases with relatively low or relatively high levels of this microRNA. And this showed that dozens of mRNAs are downmodulated in CLL cases which have high levels of this microRNA. So those are all the genes in blue here, including 2 genes that have evolutionary conserved binding sites for this microRNA, GAP1 and FOXP1. And I think this approach allowed us, this unbiased approach, allowed us to identify targets that we would otherwise not anticipate it, and also I think, prevented us from falling into the trap of following some of our favorite molecules. We further validated that patients that have high levels of miR-150 indeed have low levels of both of these proteins, as you can see on the Western blot, and transfection of cells with synthetic microRNA downmodulates the levels of GAP1 and FOXP1. Importantly, both of these proteins regulate BCR signaling, as evidenced by an impaired calcium flux after silencing of these genes with siRNA in CLL cells. So this is the model that we proposed, where miR-150 regulates two targets, GAP1, which is an adapter protein that connects BCR signaling with PI3 kinase signaling, and a transcription factor, FOXP1, that affects the phosphorylation of AKT after BCR cross-linking, indirectly, probably through regulating a number of genes involved in BCR signaling pathway, and I'll get to it later. We also recently understood how the levels of miR-150 are regulated in CLL cells. We showed that T-cell interactions or BCR signaling that leads to MYC activity directly represses miR-150, and that's because MYC is directly binding at least two sites in the promoter region of miR-150. And George Kalin's group recently showed that this microRNA is very constantly downmodulated during the transformation of CLL into Richter's, and I think it's likely that this is because of the frequent gain of MYC activity during this process. And we actually saw exactly the same thing in the transformation of follicular lymphoma into DLBCL that gains MYC activity. So in the second example that I would like to show you, we noticed that one of the miR-150 targets, the transcription factor, FOXP1, is downmodulated in CLL cells that are treated by FCR in vivo, so there is DNA damage going on. And we showed that this is p53 dependent, since artificial stabilization of p53 with a small molecule downmodulates FOXP1. And this actually prompted us to study the connection between p53 pathway activation and BCR signaling, which, to my surprise, is something that is quite understudied in mature B lymphocytes. And we showed that a small DNA damage that is actually not causing any change in cell viability affects the responsiveness of CLL cells to BCR cross-linking, as evidenced by phospho-ERK levels, phospho-AKT levels, or calcium flux signaling. And we thought that maybe this stems from some natural mechanism of B cells, normal B cells, that actually need a way how to stop the pro-proliferative signal from the B cell receptor when their DNA is damaged. We initially thought that it's going to be quite simple and that the miR-150 is actually induced by DNA damage and it's repressing FOXP1, but that was quickly excluded. So this microRNA is not affected by p53. But there is another microRNA called miR34A that is very rapidly and dynamically induced in cells under p53 stabilization or DNA damage. And we showed that miR34A is a very potent regulator of FOXP1 translation, approximately 2 times stronger regulator than miR-150, which I think is in line with actually FOXP1 having 2 binding sites for this microRNA as opposed to 1 site for miR-150. So to cut a long story short, what we think is happening is that p53 gets stabilized during DNA damage response. It activates miR34A, and that microRNA downmodulates FOXP1. And we showed that FOXP1 is a natural repressor of CD22. So during DNA damage, the transcription factor goes down, and CD22 is upregulated. And CD22 serves as a docking site for phosphatases on the cell membrane, as you can see on the left, such as P10 and SHIP1, and that allows the cells to stop BCR signaling. So here you see that CD22 is approximately 2-fold increased after DNA damage in CLL cells. I think it's not surprising that CLL patients with p53 aberration have the lowest levels of miR34A, since p53 is directly transcriptionally activating this microRNA. However, there is approximately 5% of CLL patients that have relatively low levels of this microRNA, and we still don't know what is the mechanism for that. We tested some mechanisms that were proposed by others and maybe some relationship to the 13q deletion. We didn't see any association with 11q deletion. So probably the most likely explanation is that in these patients, the gene for miR34A is heavily methylated, which was suggested by other authors. Anyway, we were able to define specific cutoff values for low levels of miR34A that independently associated with a very short overall survival of CLL patients of less than 2 years, independently of the p53 status. In my third example, I would like to show you a study from Tom Kipp's lab where he took advantage of the CXCR4-CD5 model introduced by Nick Chiarazzi. He sorted the CXCR4-DIM CD5 cells that just left the lymph nodes and showed that these cells have 5 to 6-fold higher levels of miR155. And this microRNA acts as a negative regulator of a phosphatase SHP1, which allows these cells to actually have more active BCR signaling, so they're more responsive to BCR ligation with anti-IgM. So here is the model where they show that in the context of lymph node microenvironment, which is reflected in the CXCR4-DIM CD5 subpopulation, the miR155 is high and SHP1 is low. And in another report by Pablo Pezzo's group, they showed that in a very similar context, induction of miR22 is repressing the second important phosphatase, P10. So in the context of tumor microenvironment, both of these microRNAs are upregulated, which allows the cells to have more active BCR signaling. And both of these microRNAs are regulated by T-cell interactions. Here I'm showing the example of miR155, which is induced by CD40 ligation. Also, in general, the levels of this microRNA are much higher in CLL compared to normal B cells. And we have shown, we think that this is because a CpG island in the promoter region of this gene is differentially methylated, and this whole region is transcriptionally active. And this data and other data also led to a proposition to conduct a clinical trial with microRNA inhibition. And that's a phase 1 trial that is run by a Mraz company where they're blocking miR155 in CLL and diffuse large B-cell lymphoma. And that's basically a completely new class of drugs because it's based on either mimicking or, in this case, inhibiting microRNAs. There is also an interesting report suggesting that maybe BTK itself, so key kinase in BCR signaling, is directly regulated by microRNAs. And there's a couple of microRNAs that have predicted binding sites in BTK, and for several of them, including miR210 and 425, it was shown that transfection of the cells leads to downmodulation of BTK levels. Both of these microRNAs can be induced by HDAC inhibitors, which you can see in the middle. And also this HDAC inhibition leads to downmodulation of BTK in CLL cells. I actually don't know how regulation of BTK by these microRNAs is relevant in natural biology of CLL, but it might have some implications in the use of HDAC inhibitors. So in the last part of the talk, I'll like to show you that CLL cells not only use the microRNAs to regulate BCR signaling, but actually also CLL T-cell interactions can be regulated by microRNAs. And we again used the CXCR4-DIM CD5 model, and we sorted the cells that exited the lymph nodes and showed that they have low levels of all three members of the miR29 family, miR29A, B, and C. And this is interesting because it's for a very long time known that low levels of these microRNAs associate with shorter survival in CLL. So here it's our data, but others have seen the same thing. And using the techniques that I have described before, using gene expression analysis, we identified a potential target of this microRNA that is anticorrelated in the context of the CXCR4-CD5 model, a molecule called TRAF that has high levels in the instance where miR is low. And indeed, CXCR4-DIM CD5 bright cells have high levels of TRAF protein. We further showed that transfection of CLL cells with a synthetic miR29 really robustly affects TRAF levels in primary CLL cells. Actually, all of these three microRNAs have identical seed sequence, so they compete for the same binding site in the mRNA of TRAF. So kind of all their levels altogether determine the level of TRAF. And what I think is most important, we showed that the TRAF levels are determining the responsiveness of CLL cells to CD40 ligand produced by T cells. So here you can see the transfection of CLL cells with a miR29 mimic and how it affects the response of these cells. So in the model that we propose, BCR signaling leads to accumulation of MYC, and MYC is repressing miR29, and I did not show you the data, but this was shown 10 years ago that this microRNA is directly repressed by MYC. So the low levels of miR29 allow for accumulation of TRAF4 in the context of microenvironment, and that allows the CLL cells to become more responsive to T cell interactions, especially CD40 ligand, and have bigger NF-kappa B activation. Given what I said about regulation of this microRNA, I think it's quite logical that the levels of miR29 are induced by ibrutinib, because ibrutinib is blocking the MYC activation through B-cell receptor, and the target molecule TRAF is down-modulated. So I think this suggests that it might be an indirect mechanism how ibrutinib is actually affecting the CLL T-cell interactions. This is my final slide. There won't be any text. What I tried to do here is I summarized the 6 microRNAs that we know are regulating BCR signaling in CLL. I did not have a chance to talk about miR18 and 19 because that was mainly studied in diffuse large B-cell lymphoma. And on the right side, I also included the miR29, which I think is connecting the BCR signaling with the T-cell response in CLL cells, and the CLL cells are using it in this way. And in orange, I have highlighted microRNAs that are considered as potential therapeutic targets. So there is the trial running with miR155 inhibition, but we heard an hour ago about miR29 being delivered through a ROR1 antibody, and there are other potential candidates. I would like to also mention that for the long non-coding RNAs, we don't know. We have no idea how those are regulating BCR signaling. And I think philosophically, those 200,000 long non-coding RNAs are going to keep us busy for probably the next decade at least. Thank you for your attention. I would like to thank great collaborators, especially, of course, my lab members, but also collaborators, Tom Kipps from UCSD, and clinicians and collaborators at University Hospital in Brno, especially Sharka, who's sitting here. Thank you very much, and I'm happy to take your questions.
Thank you, Marek, for a very interesting presentation, and we have 2 minutes for discussion and your questions, so if you have any, please come to the microphone.
Thank you very much for your excellent conference. My name is Besares from Argentina. Recently, Manzoni et al. Have published a very interesting paper about metformin and regulation of DCR and his action about the stabilization of several microRNA in several cancers. Do you know something about this item?
I'm sorry, I cannot comment on that.
Metformin.
I haven't read that paper, so I'm sorry.
Thank you.
Any more questions? If not, thank you very much once more.